Okay, so my presentation today is laser surgery, a powerful tool in biology. So with this presentation, I want to show you a bit all the parameters that normally you have to be aware of when you want to do laser dissection. So which kind of laser and which property of laser you need, and then what kind of optics and things that will allow you laser ablation. I will show you that with different laser, in fact, you can do different things, different type of laser dissection. And then finally, I will show you different examples of how you can then implement this technique on different in vivo specimens. So this first video that I'm showing you is something that I published myself in 2007. It's a work that I will present you in the end. So what you're seeing here, in fact, are cells, and you can see the outline. And this is the apical cross-section of these cells, of a tissue which is much bigger. In fact, this tissue, it's the ectoderm of the early developing Drosophila embryo. And what is marked here is epithelia caudine, okay? So, which are proteins which are responsible for the adhesion among cells. And so this is a very interesting experiment. It was quite challenging at the time. And the idea was really to measure, in fact, tension along junctions. So to be able to dissect actin, which is along this junction. It's called junctional actin. And then see what happened. In addition, you see that there is also other type of sources of lasers. In fact, at the beginning, there's an ablation at the junction, but then you see then there's also a laser that is shooted within one cell and then within the other cells and more times, okay? And also you see when the laser is shooted within the cell, you see the cell becoming fluorescent, becoming bright, okay? And this is in fact a control technique that I was using. I will present you also after that. That controls, in fact, the state of membrane. How is the membrane broken or not? Okay, and then I will explain you how I did this. So every time I will show you laser dissection, I will always put a table underneath. It's like a bit boring table, but it's a very necessary table in which, in fact, is specified exactly what kind of a laser with which power I'm using, which objectives I'm using to, in fact, do this kind of laser dissection. Which is something that normally sometimes when you read the papers you do not find, so you don't know how people did this. And it's very important to know how they did it to be able, in fact, to judge their experiments, okay? So here you will have the type of process. The first case is ablation, the one in the center. The second one is photon caging. Then here's the wavelengths. In this case, it seems to be the same laser. The pulse duration of the laser. Pulse repetition rate, average power, exposure time, the objective that I use, the objective transmission at that wavelength, then the pulse energy and number of pulses. This is kind of the table you will find every time you will see. And sometimes you see a question mark because I could not find it, in fact, in the reference paper. Yes? I will show you at the end. During my talk, I will specifically explain how it works, as just to present. So this is showing that, in fact, when you send light on matter, you have many processes like reflection, you have scattering, you have also part of transmission of this light. But part of this light, in fact, is transfer, in fact, to the material. And this is the absorption. And this is what we are interested in. So it's very important to choose, in fact, the incident light when you want to do laser dissection. So what we normally use, it's laser sources, mainly for two reasons. One, because they are spatially coherent, and also temporally coherent. Spatially coherent means when I shoot this laser, it's a small spot here and it's also a small spot there. In this way, I can direct my laser towards different entrances that could be into the microscope. So it's a light that it's easily you can manipulate it, okay? And this is, then there's the temporal coherence, which is the fact that it has different colors in the spectrum. And you will see that different colors will, in fact, react in different way on the specimen. So there are four main laser parameters that you have to take into account when you want to do laser dissection. The first one is power, and you can understand why. The second one is the spatial compaction of photon. Okay, so that means that if this laser, now I focalize it into an objective, and this will be highly focused, then the intensity will rise. And the fact that you can rise this intensity, you're spatially compacting the photon onto a small surface. Then you can join the first 2 points in one parameter, which is called power density. It's exactly the same thing. Or if you want, intensity. It's just the same thing. Then we have temporal compaction of photons. So now it's no longer in space, but it's in time. And you can send, in fact, clusters, packets of photons by compacting them. And this is what distinguishes what is called a continuous laser to a pulsed laser. Then you have the wavelength. So we talked about the spatial coherence. And then finally, the pulse repetition rate. This means that in the unit time, how many pulses does the laser send? This is the case of pulsed lasers. So I will go through each of these parameters and explain them to you, okay? So if we start to take into account the first two, power density and temporal compaction. So there's a diagram that you can draw. This is a diagram that has been drawn by Nims in one of his books that kind of recapitulate much of the work that has been done in laser dissection. So what you have here, it's pulse duration. That means it's the width of the pulse in time. That means how much are the photons compacted in time, okay? This would represent, for example, microseconds, nanoseconds, femtoseconds. You heard about femtoseconds laser mostly when you think about this two-photon process when you want to do two-photon imaging, okay? So it's very interesting to see that you find different regions of interaction. You have photochemical interaction. One photochemical interaction I think you know very well, it's photosynthesis chlorophylliana. So it needs, in fact, a wave which is almost continuous, no pulses. Okay? With not a lot of power density, which is the intensity of the light. Then, if you lower, in fact, the pulse duration, what you have, you start having, in fact, thermal effect, one of which I will talk about is carbonization. If you higher, again, you have photoablation. So photoablation is written here. In fact, you're like breaking the bonds between molecules. This will increase locally the volume, and the molecules, in fact, will be etched. Normally, this is used, in fact, for surface etching. Then another technique which is much used now, mainly for in biological sample, it's called plasma-induced ablation. So the plasma-induced ablation is different because what it does, it creates a plasma. A plasma, it's ionizing the matter. That means that, in fact, you're detaching all the electrons that will create an electron cloud. In this way, you will locally, in fact, destroy the matter. So what I took here is just a video from YouTube that will show you a plasma. Because, in fact, what is interesting about plasma is that the plasma can also emit light. And also, it can make a mechanical wave, so it will emit sound. So the sound, you will hear it from my computer. So this is a huge, big laser, you can see. But you don't need such a laser now to make a plasma. And it will make a plasma in the air. So what they will do, they will just put a lens in front of the laser, focalize the light. So it will increase the power density in a way to get to the plasma threshold. So this sound, it's not my computer, this is the sound of the plasma, in fact. And you can see there the small plasma which is formed in the air. Okay? This is also what normally we now use to do laser dissection within, for example, embryos.
The frequency of that sound is repetition rate.
The frequency of that sound, yes, it's also dependent on the repetition rate. In fact, I had the live experience to see this when I was in Fresnel, in Mosaic, which is an institute of optics and also biology. And they had, in fact, a laser which had a much lower repetition rate. So it was very slow shocks, like, but it's exactly this. So it's the repetition rate. Then finally, what you have is another class which intersects also the plasma-induced ablation, which is photodisruption. Photodisruption represents the presence of a plasma, but in addition, you have shock waves and bubbles. Normally, bubbles are created by the fact that locally you're vaporizing the matter, and this, in fact, will increase the volume. This bubble can increase and then implode. So there's an explosion and implosion process. So for the photo disruption, I wanted to dedicate one slide, because it's a very nice work if you want to look at it. It's also done on Drosophila, and this was done in 2007. And it's very nice because they use an hydrophonic needle that in fact will feel the formation of the bubble and then the implosion of the bubble, because this will create in fact mechanical shocks. It's a needle. It's a needle, hydrophonic needle. So in fact, it's a needle that can feel the mechanical waves. Yeah, so it's a very nice experiment. And they've been using, in fact, so it's very interesting that every time I present you this, you look in the table and see what kind of a laser did you use. So we can see that they used, in fact, for the first experiments, a 355 nanometer, which is a UV laser. And then for the second one, they used a green one. And the pulse duration was 4 nanoseconds. And here you can see the bubble oscillation period, and on the other side, in fact, the bubble radius for different cases. They did it in water, and then they also did it in the Drosophila embryo. So this confirms, in fact, this experiment that normally when you have lasers, they have pulse duration around the nanosecond range, which means very wide pulses compared to the femtosecond range, or picosecond, femtoseconds. Normally the dissection, so the laser ablation you have, it's always accompanied with shock waves. Because what happens is that your photons are so much spread in time that you have to higher, in fact, the power to reach, in fact, the plasma threshold. In that way, you will ablate, but at the same time, all the photons which are remaining will create, in fact, surrounding destruction. So will create shockwave or plasma or bubbles. So in the case of the 355, they can still detect bubbles, which will depend then on the pulse energy they will put. While on the 532, which is a wavelength which is much more energetic than, is less energetic than UV, that means they will have to put even more power. This will create even more damage. So they will have to put more power to reach the plasma threshold. Okay, then I wanted to show you this other experiment, which was done in DevCell in 2009. And if you look, in fact, they are quite using more likely the same laser. It's a 365 nanometer laser. And it's 3.5 nanoseconds pulse duration, between 3 and 5. And now if you look, in fact, at the pulse energy they use around 60 microjoule. You do not specify exactly where they measure this power, but this is what they say. And if you compare to what they use here, it's 1.22 microjoules. So you see they use a pulse energy which is much higher, but more likely than the same pulse duration and wavelengths than this first experiment. So this is very interesting because this work wants to study, in fact, the tension along junctions, okay? But there's no control of, are there formation of any kind of bubbles? Are there formation of any kind of shock waves, okay? So I can show you this movie, but from the movie, in fact, you cannot judge much. You cannot say. So what this work normally should need is, in fact, is controls. So what is happening now is that this laser dissection, I remember at the time when I started doing laser dissection, it was not much used. So many people were asking about what are you exactly doing? But it seems now that laser dissection is widely accepted. So people do not, in fact, ask with which laser are you dissecting. So which control should you have to be sure that there are not collateral effects? So this presentation is also to make you more critical when you have to review a paper when they are using this kind of a tool. Okay? Yes. This is exactly one set plane. Yeah, so yes, so the idea, I can agree in the sense, by looking at just by eye, I feel already that this experiment needs controls. In the sense, I feel that there's much also extra damage. And also, they specify in the paper also, I don't want to talk too much about this, I want to go on, but there's additional collateral effects, local bleaching, blackening of the vitelline membrane. So definitely also there's much, set effect, not only on the plane. So I don't want to go too much into this paper. It's just to tell you, send a message out. When you have a paper and they are using laser dissection, you have to be critical. So what are the main parameters you have to see? You have to look to judge the work. Okay? Okay, now there's always many people that ask me, yeah, but it's better UV, is it better infrared? So it's not that one is better than the other. It depends on the question you want to do, or you want to ask, and then on the experiments you want to do. So here, the main difference between UV and infrared when you want to do laser dissection. So UV, as you know, it's a photon which has more energy. That means that to send an electron from this bound state to its ionized state, you just need, for example, one photon. If now instead you want to use the infrared, you will need more photons. So on the first, if you look at it like this, it would say, okay, then UV probably it's better because you need less photons. So what happens is that then the absorption with the UV will be linear, okay? While in fact, with the infrared, if you will send 1, 2, 3, 4, nothing will happen. You have to go up to 5 at least to see an effect. So in this case, it will become threshold. In this case, with the UV, then, if you have a uniform absorption, what you will have, in fact, is that when you will send the UV on your specimen, not only the UV will be absorbed on the focal point, but also much before and probably also after. This is instead. Well, if instead you take into account the infrared, you will see that what happens is only where you have the maximum intensity, thus where you compact spatially more photons, is where you will reach the threshold and thus dissect. So what happens is that with infrared, it's much easier to do bulk dissection within tissue. With UV, I think it's more suited for surface dissection. Okay. So the other parameter I talked about, if you remember, is the repetition rate. So in the unit time is the laser sending. So this is an example where in fact the laser is sending kilohertz pulses. So what happened is that after a pulse is sent, there is some energy which is deposited that will produce the ablation, but also there will be also some thermal effects. And these thermal effects, so there's energy which is deposited. This energy, in fact, will go down with time, will diffuse in the tissue, before, in fact, the other pulse will come. So the pulses will one from the other. So you will not have other effect and just the effect of what each pulse normally will do. If now instead you start to send a train of pulses, what you have is that you will have this energy which is deposited doesn't have the time, in fact, to diffuse around the tissue. So in fact, this energy will start to increase. So as I said, it's because of many things, depending on which laser you use. That means what is the pulse duration and what is the power you use. So if it's, for example, a femtosecond laser with a certain power density, it will be plasma. If in fact you're lowering in fact this pulse duration, or you lower the power, then will be temperature based. If you have nanoseconds and you have another type of power density, then will be hatching photoablation. So there's not one type. It will depend always on the table that I will show you below. So you will look at the parameters, you will know, okay, given this parameter, I will know this type of ablation will be of this type. And you will know also that each type of ablation will have more or less collateral effects, and which collateral effects. So this is the kind of information you kind of have to be critical and say, okay, this paper is using this tool, does he need to do this control or not? Okay, then this is another very important graph, which is showing how the objective is important, and more specifically, one parameter of the objective, which is the numerical aperture. That means how much, in fact, the objective can focus this photon on one point. Since he will focus these photons, the power density will go up. Okay, given a certain number of photons sent by the laser, the power density will go up, thus the threshold energy to reach the plasma will go down. So it's very important to have an objective which has a very high numerical aperture. In fact, during the workshop, for the people that did the workshop, you will see we always used objectives that had numerical aperture up to 1.3 or 1.4. These are the ones which are best suited for this laser dissection. Okay, So let's pass to biology. Now I'll give you some examples of how this can be applied to biology. So one first example that I like, it's a work by Kumar et al. So he has, in fact, cells, which are in apertures, living cells, and he can see actin fibers. And what does he do? He uses, let's see what kind of a laser. It's an infrared laser. And it's a femtosecond laser. So it's 100 femtosecond. That means the pulses are very, very sharp. So in time, photons are very much compressed. And this, all the other. Then the numerical aperture of the objectives, as you can see here, it's 1.4. So it's a good objective to do this kind of a laser-tissue interaction. So the Ablation does it there where the arrow is pointing. And you will see there's a fiber, and then you see a hole, which is in fact expanding. And in 2006, in fact, Kumar was asking himself, what is really happening? Is there a retraction of the fiber, or is there a depolymerization of the fiber? Because from this imaging still, it was not clear. So he went on and he found, okay, this is again another experiment he does on a thicker fiber. And what he does a punctual fiber, and the fiber is cut only in the inside. And then he sees like a ring inside that is expanded. But still, this could not show if the fiber was really stretching or if it was just a local depolymerization of the fibers which were inside this bundle. So is there retraction or disassembly? This was his question. And I think he kind of answers in a very smart way. He tries to say, okay, let's see if I can find some kind of a case like this, where I have a bifurcation. So if I manage to cut exactly there, in the intersection, well, if there's disassembly, then all three will start to disassemble, and I will start to see, in fact, the edges of each fiber. If there is not disassembling, two will stay connected, and one will detach. So this is what he shows here. So you have to have very good eyes to see that. But if you look, this fiber here, it starts from here, and then right here, it's divided into one up and one below. And here's exactly where he will do the dissection. And then you will see here's the dissection. You will see in time that this part above, it will stay exactly like this, showing in fact that most likely you have retraction. Possibly still disassembly, but certainly retraction. These fibers are attached to, I think, the extremities of the cell. And they are then attached to the focal point. You know, I think this cell, I don't know, it's zoomed in, but I'm sure this has an extracellular matrix. So it's certainly there are integrins, thus is attached to it. And then on the focal points of attachment, these fibers will join to these focal points. In the same way, he uses also another technique. So I like to show you this technique because in case you want to use this laser, these are very useful technique to understand what is going on. These are kind of a controls. So what he does, in fact, he does photo bleaching on one part of the fiber, and then he decides to cut the other part. And so what he sees is that this photo bleached part, in fact, moves also. So clearly that means that certainly you have retraction of the fiber. Okay? So the retraction is certainly present.
It doesn't move without.
What?
The fiber is just getting depolymerized. It doesn't move without the cutting.
No, right here, what he shows is that the cutting certainly produces fiber relaxation, for sure. He doesn't show that there is, and also with this one, he kind of shows that there is no disassembly. Right? So with this, he shows that there is no disassembly, so this is wrong. And with this, he shows that this is correct. So the time scale, okay, it's not written here, but I would say it's the term, yeah, like around half a minute or even less. So then it's very nice because what he does, he follows the hole which is formed, and then he kind of fits this with naively a bit with a viscoelastic model, and it seems to fit well. And then he starts to measure different quantities, for example, the final length of the fiber, and then also the constant, which is represented the Young's modulus. So this is the ratio between the elastic coefficient, the viscous and the elastic. And interestingly, what he shows that this tau seems to increase for thicker fibers, okay? Thus, he thinks that probably there's some viscous property which is being enhanced. So what he thinks is that for thicker fibers, probably there is an interaction effect among bundles. And this will make, in fact, the system more viscous. While for thinner fibers, it seems that a bundle, in fact, is acting like each fiber independently, one from the other one. Then what he does some inhibition of motor protein, in this case of myosin, by using ROCK inhibitor and MSTK inhibitor. And what he sees that compared to wild-type effect, the relaxation is much slower. Finally, he tries also to do this by looking at the full cell and putting it, in fact, on a gel with, I think, beads below. And by looking at the displacement of the beads and knowing, in fact, the properties of the gel, he will calculate the traction force. Okay, this experiment is that it's another one. This is, I did at the times, I think in 2007, and it was in collaboration with Thierry, that came, in fact, in my lab when I was doing my PhD, and he asked me, Matteo, can you try to do something with these cells? Because I'm creating micro patterns with a laser. So he creates a pattern that has adhesive properties of fibronectin. So cells, in fact, they can spread on this and they can adhere on this. And depending on the shape he can create, cell will make different shapes and also create different cables of fiberactin. So he asked me really to try to test different type of cable inside and outside cables. So this is just one example. For example, what I did a dissection on one of the greatest cable in this cell. And it was interesting, in fact, to see what was the output. So I would like somebody of you that would imagine what is the final output. What kind of a shape will the cell take? So what do you imagine? I would like somebody of you to give his opinion, or her opinion. Do you want to say something?
Would it round up?
We think it will round out completely, so it will retract back, or will it expand? It will retract? Okay. It can become linear. But in general, the idea is, would it retract or would it expand? You will expand? Okay, it's interesting because you see, retract and expand. So we see biologists in a very different way, I think. What else? Any other opinion? So this cable, I think, is completely cut. And it's a cable which is really at the periphery. So it's not one of the internal cables. It creates the dome, the architecture inside, but it's more at the periphery. So the others do have no opinion. Expand? Okay. Expand. You want to say something? Okay. Okay, we have 3 people of 4 that have an opinion. Okay, we'll play it back. I was also quite impressed because biology is very surprising. So very interesting. In fact, he was right. And that's why now I will put the title. What is very interesting is, in fact, is that the retraction shows really that these cells are quite complex. And how when you look at contractility of a cell, you cannot forget that this cell is, in fact, is anchored to something. And these things are very linked to one another biochemically. So you have to take this into account always when you do your experiments, okay? Okay, now I will try to present you a work which is more...
Sorry, the action is can depolarize or you're seeing polarization?
Okay, in this case, I didn't do this control with a mark, right? But I did other experiments exactly like this by doing exactly like Kuma did. So this is really retraction of the fiber. So the fiber was definitely cut, and definitely this fiber was retracting. I cannot tell you how much this was depolymerizing. But definitely the fact that we're releasing tension from these edges is changing, in fact, the adherence of the cell to the extracellular matrix. So it's doing something there. So you know that tension, it's needed somehow to maintain, in fact, the focal adhesions. So it's much more complicated just by saying, okay, I cut an elastic, let's see what happens. Because then you have to take into account, in fact, other biochemical reaction happening at molecular scales. So we have no vision of the membrane. At the time when Manuel came, he didn't have any membrane, so he had actin and this fibronectin. I think this was probably the only line at the times he had. So I don't know exactly what happens to the membrane, but I would say also the membrane probably would retract with the. So here probably we see still a bit of probably the membrane. I don't know. Probably the actin retracts, the membrane maybe stays still a bit attached. Probably. This could be. You see, if you see a bit there, there is some green stuff there still. Probably. So we did other tests. So in this one, in this case here, we did not spend time to do any control on the membrane. But it's true that when we did break the membrane, we saw many leakages, and in fact there was much flux of the cytoplasm inside. So normally by doing this experiment like this, we were quite sure that there was no, at least, major membrane disruption. Maybe little membrane that was then healed, in the worst case, but it was not major membrane disruption, because the cell would just empty out. There is, wait. On these ones? Probably not. Probably not. Yeah, this could also be. This could also be. The problem is that when you break, it depends how much you break the membrane. If you disrupt the membrane a lot, then you will see that there's no more cell at the end, because the cell is emptying out. So more than calcium going in, there's cells going out. Okay, now I'll present you instead in a more complete way a work that I've been doing, that I've done in 2007. And this is on a live embryo. This is the early developing Drosophila embryo. And what you see here is many hexagons. And this hexagon, in fact, it's a specific view of cells. Okay? So each hexagon is a cell, but in fact these cells are very long. They're like 30 micrometer long. These are epithelial cells. So the Drosophila embryo at an early stage is made of one epithelial tissue of cells. And these cells at the apical surface, which is the one that you're looking at, they have adherence junctions. And this is what you see, because this is something marking epithelial adherence. Okay, it's not written, but this is what it is. So this work has been done by simplifying this tissue in a 2D tissue, as you see it now. So we forgot completely that, in fact, these cells have a 3D shape. Okay? So this is some kind of a simplification. So what you see, this tissue, what it does, it kind of flows down, but then what it does, it elongates. Okay? So for now, we will just forget about this downflow, and we'll just think about this elongation. If you will be there on the 21st, I will present also. You will see instead that I'm very interested in this downflow, and I'll forget about the elongation. But for now, we take just into account this elongation. So this elongation has been studied a lot, and it has been shown that this elongation, which is called germ band extension, so it's the extension of this tissue, it's possible because what is happening is within the tissue, cells are going through a process called cell intercalation. So if you want to understand what is cell intercalation is about, you can take a piece of tissue like this and zoom in, and now you can color the cells. You can color one row in blue, then one in green, and then the one below in red, in magenta. And then you look 30 minutes after that. So you will see that, for example, here, a cell blue, normally, it's in contact with two green cells, and also the red cell with two same green cells. But after 30 minutes, this cell here, you will see it's in contact with the red cell, the magenta cell, while the two green cells now are separated. So this intercalation, in fact, it's an exchange of neighbors. Okay? So what happens is that many neighbors are exchanges, and as you see, this piece of tissue, which is exactly the same now, it's elongated. So how does cell intercalation happen? So how do cells exchange their neighbors? So if now you focus on one junctions, and thus, for example, these four neighbors, what you see is that at the beginning, this junction puts in contact these two cells, but then a junction shrinks in a punctual junction so that 4 cells come in contact. And finally, there is a new junction which is formed that puts the cell on the top in contact with the cell on the bottom. So very interestingly, in this work by Berthe et al. In 2004, what Berthe shows that this process is an active process. So cells locally, what they're doing, in fact, they are shrinking their junction. And they're shrinking this junction in a polarized fashion, because all the junctions which are shrinking are not this junction, but are all the vertical junctions, which are junctions which are parallel to the dorsal-ventral axis of the embryo. So there's some kind of a polarity within this process. And by looking at myosin 2, this is myosin 2, marking myosin 2, you will see that you have this polarity. So junctions which are vertical are the ones which are more enriched in myosin 2. So she proposed a model where, in fact, myosin 2 is acting like it is a motor protein. So it's not acting as a cross-linker as it could also act, but more likely as a motor protein. And it will produce, in fact, forces that will reduce the junction, thus initialize the intercalation process. So my work was to try to test this model and to see if really there were greater tension along this vertical junction. And this is what I did. I did laser dissection, and this is why you see again this table, because every time I will show laser dissection, you will see all the information below. So again, what is it? Is it an infrared laser? In fact, it's a neodymium YAG solid-state laser. Again, it's a femtosecond laser. And this is the kind of objective that I was using at the time, water immersed. This is the pulse energy. Okay, what you see here is myosin actin binding domain. So it's a way, in fact, to visualize actin in the Drosophila embryo. And the yellow arrows are pointing out one junction, one vertical junction, and then the ablation is pointed out with the red arrow. And what you see, in fact, you have depletion of actin at the junction. Okay? And this is just a Kymograph. A Kymograph is a way, in fact, to show with one single image what is happening in time. Okay? It's just showing the junction at this minus 5 seconds, which will be correspond to the first line, and then the same junction, each pixel on this axis will represent time. Then we looked at epithelial cadherin, and we did the same experiment, and also in this case we saw the depletion of the cadherin. So this was a very nice experiment showing that there was interaction between actin and cadherin. There was a tethering interaction. And that most likely what was happening at the junction after dissection is that actin was retracting and bringing along the clusters of cadherin which were punctuating the junction. So then what I started to do is to try to measure, in fact, speed of relaxation and try to compare, in fact, the speed of relaxation of junction which had different directions. So parallel to the ventral dorsal-ventral axis, that's most likely enriched in myosin 2, compared to other junctions which normally are not enriched in myosin 2. So by measuring the speed, and by doing ablation, for example, here we considered one of the tension going down to 0, and then we decided to use the speed as a proxy, in fact, for tension. So the more you will have relaxation speed, the more you have tension locally, and the less you have tension. So we did it, and we considered to measure initial length. The length of the junction for us was a way to consider the stage of intercalation of a cell. If you had a large junction, it was like an early stage, okay? So probably less tension. And instead, when the junction was shrinking, so probably more enriched in myosin 2, then with more tension. And this is exactly what you see. A cell with larger junctions, vertical junction, you will see that it will have less tension, and then the tension will increase while the junction is shrinking. Then we inhibit also myosin 2, and this is the experiment. So the one on the left you saw already was my first slide. And then what you will see on the other side instead is a Rho kinase inhibitor. And you will see, in fact, that you will have no relaxation. So this is the case for the Rho kinase inhibitor. So we're inhibiting the motor protein. Then we did also dissection not only on the vertical junction, which are the one in red, the red circles, but also on other junctions, which normally are not enriched in lysine 2. And we could see, in fact, that there was a difference in relaxation. So the final model was really that there was an anisotropy of tension locally at the junction, and that this will allow, in fact, the first step of intercalation, which is junction reduction. Okay, so now I think I will now talk about how I controlled, for example, membrane integrity. I think somebody asked me about this before. So what I did here, as you can see, I did the ablation again between cell 1 and 2 in this spot here. And again, you see the depletion locally. And then you see a green arrow in this one here. And you see uncaging. So what I did inject in the embryo at a much earlier stage. In fact, a probe, which is that you can uncage. So normally this probe, you cannot see it. It's a very tiny probe that can diffuse very fast. So if you had a completely empty embryo full of yolk without any cells, and you would inject this protein would diffuse within the embryo in a few seconds. So it's a very, very tiny molecule. And what happens is that this molecule is present within all the cells, because by injecting it at an early stage, while the cells form, each cell will have a reservoir of this molecule. And this molecule is not visible until you uncage it, normally with a UV laser. This is the kind of laser or wavelength that this molecule needs to be uncaged and to be visible. So this is what I do. So I do the dissection, and after that, I uncage the molecules to see if there are leakages. In this case, you see that one cell lights up, but the one on the other side, cell number 2, does not light up. What I do here, I increase the power. So I do the dissection again, and by uncaging one cell, on one cell, we see also the other cell that highlights right away. So that means that at this point, the two cells are communicating. That means there was a membrane breakage. If you higher the power even more, you will see that the two cells will start to be engulfed in the tissue, and finally the two cells will completely disappear. In fact, you will see here what I'm doing. I'm increasing the average power, okay, from the first to the last experiment. I didn't measure it. I didn't spend too much time. For me, it was like a binary control. If I have membrane breakage, I will not take into account. If I do not have, then I will take it into account. But I didn't go in and analyze that more carefully. So then I went on and tried to better characterize my tool. So what I wanted to do is to try to measure, in fact, the spot of ablation. So how small was the spot? How big was the spot? So this was quite challenging, because one good sample would have been, for example, a monolayer of fluorescent stuff. And by ablating this, you could kind of measure the spot. The problem is that the samples or beads, for example, they're not biological samples. So it was very important to do this kind of a measure exactly within the Drosophila embryo to see what happens. So directly on actomizing fibers, for example. So since the junction, they're very thin, it was tough to do this kind of analysis on this junction, because in fact the junction was completely depleted, so there was no way to, in fact, to see the hole. So I decided to do the same experiments by using, in fact, the ring of actomizing, which is formed during cell cytokinesis. So this is what I'm doing here. I'm making many ablation and cutting the cytokinetic ring. So by doing one single spot, I could go down and say that, in fact, the spot is big. It's around 400 nanometers big at the most. So another type of analysis that I've done, so these are all experiments that I've been doing just really to characterize my tool. So this is one that I liked a lot. I was very willing to do this, which was to try to couple, in fact, a spectrograph within the microscope, and thus collect the light that I could probably produce by ablating. So as I said, this type of ablation, if we look, it's in the infrared and it's a femtosecond. And if we go back to the diagram as before, this is in the plasma regime. So normally it's an ablation that theoretically it's plasma induced. So the idea was to try to find a signature of this plasma and see if I was really producing a plasma within the Drosophila embryo. So it was very interesting that, so what I'm showing here in this graph below is in green, in fact, is the emission of GFP. So you have the wavelength, and this green stuff is the GFP. All the other stuff is filters that I had in there. So this one is the quantum efficiency, I think, of the spectrograph in blue. Then this is another dichroic that I had, and this was a filter that helped me, in fact, to visualize the junction. So this is, in fact, then what I collected with the spectrograph. So by sending the laser with different power, so starting with blue, then green, black, and red, you will see that at the beginning you have, in fact, an emission which really correlates a lot with the GFP. So what I was doing at the beginning with lower power, I was doing two-photon imaging, in fact. So I was exciting the GFP and collecting this one. But then by increasing the power, what I could see, in fact, that there was a threshold level over which, in fact, the intensity that I could collect was much higher. And this is also showing in this graph. At the beginning, it's linear with power squared, which is the two-photon, in fact, emission of the sample. And after a threshold power, I could see, in fact, that this was much more enhanced. And by releasing this filter, I could see that also the spectrum was much wider. And this is what normally you have when you have a plasma. Normally this plasma is producing sounds, as we heard before, but it also produces lights, as we saw in the small movie. And this had a much broader spectrum. So by doing this laser ablation, I was really creating locally a plasma and dissecting the junctions. Okay, this is another work that has been done by Solo and al. In 2009 that will show you other ways to use laser dissection. So Solo, he was interested in a process.
How small is that?
So at most it's 400 nanometers.
At the most?
At the most, yes, because it could be that I'm still bleaching on the borders, because normally you have a Gaussian-shaped beam. So I cannot say how much am I bleaching within the spot. So at most, the one that I measure is 400 nanometers.
The damage is restricted. The plasma is very, very small. It's 2 tens of nanometers.
The plasma? This will depend on the power you're sending. It could be very big. It depends really on the plasma. For example, it really depends. Depends how much power. The plasma itself will go through optical breakdown. That means that the plasma will start to absorb photon and become big. So when you start to create a plasma seed, in the end it's no more the tissue that will absorb the light, but it's the plasma itself that will start to absorb the light. This will make it grow if you continue to send pulses.
The duration essentially duration and the
yes, also the repetition rate of the laser, and then how much you will expose the sample. This will determine the life of the plasma.
What's the smallest region that you can ablate?
So as I said, at most 400 nanometers.
Smallest?
As I said, the smallest, as I know.
Smallest is 4.
Exactly. Maybe it is smaller, but I do not know if I can make it even smaller. I know that the smallest at most is 400 nanometers. From the what? That's a bit complicated, because the cells are not only these squares that you see on top. It's very long cells. So you need to do, yeah, you could measure somehow the volume. Probably you could sort this out. Yes, could be another way. Okay, so I present you this other work that also at one point Solon decided to use laser dissection. This was done at the MBL with Damian Brunner. So Damian Brunner was very interested in studying the process of amyothyrosa contraction. So the Drosophila at an early stage had different tissue, one of which is at dorsal side. And this tissue, it's an accessory tissue because it will be there during this germ band extension we saw, and will play some role probably that still has not been studied. And at the end, what it will do, in fact, this tissue will contract so that the limb of this tissue, which is the ectoderm, and on the other side, they will start to match. Okay? In the end, this tissue, the amniocerosa, will die, so will produce no organ. That's why I call it accessory tissue. So what it does, this tissue, if you look at it, so what you hear here, you have the margin, this is the ectoderm, this is the amniocerosa. This tissue, these cells, they oscillate, okay? And Solon have been looking at this oscillation and how this oscillation correlates. So he decided to do laser dissection in this case. Like here, right here, you could see. And the idea that he had, it was not really to measure tension, also because the dissection here seems quite abrupt. But the idea was to block few cells from oscillating and see how the other cells around started to oscillate. And as you could see is that by ablating two cells, so destroying two cells, also the cells around, which were mechanically coupled, stopped oscillating, by showing that this oscillation was some kind of a process which is, in fact, driven by the interaction among cells. Okay, another work, more recent, by Meyer et al. This was done in Max Planck. This is on the C. elegans. So what she does, in fact, she dissects an actomizing meshwork of the C. elegans embryo at the early stage, where the C. elegans is still one embryo, one cell. What happens is that in this meshwork, a sperm normally comes in from one side, and this will create a meshwork which has some anisotropies, which will be much denser on one side and much less dense on the other side. So what she will do, in fact, she will use laser dissection to characterize the properties of this meshwork on the anterior and posterior sides. So it will do it in different cuts, on vertical and longitudinal cuts. And like this, in fact, she will show that this anisotropy of tension is the one, in fact, that will drive this meshwork, in fact, to move on one side. Even more recent work done by Berndt et al. In 2012 in Carl Philipp Eisenberg group. It was on the zebrafish embryo. So what you can see here is a process called epiboly, where in fact the tissue starts to cover the whole embryo from the animal to the vegetal side. And I think this movie you have myosin 2, which has been marked, or myosin 2 or acting probably. So he was very interested in trying to characterize this ring of actomyosin, which was in fact at the border between the yolk area and the tissue area. So again, in this work, laser dissection was used. And what he does laser dissection in this region here, which is exactly at the border between the two. Right here. Okay? And this is what you will see here on this small movie. He will do two different types of dissection. He will do a vertical dissection, so like this. It goes along the animal vegetal pole, and then a transverse dissection. So this experiment was very interesting because in the end, these were the results. So the results were that at different, in fact, stages of development, so at different epiboly coverage, what we will have is that the vertical cuts normally will relax much faster by saying that there's, in fact, much more tension along the ring than along the animal vegetal pole. But what happened is that he will try to create a model and see how much this ring, in fact, could contribute for the movement of the ectoderm. And what he saw is that this ectoderm, in fact, this component here, which is the animal vegetal pole, it's much more than what he could, in fact, predict with this model. So this laser dissection experiments kind of helped them to understand that the contribution of the ring could not be the only one, because the relaxation that they could have here was much greater than what the mechanical model could predict. And that's why he will look for other actors that could help, in fact, the movement of this tissue downwards. And he will see that there is an actomizing flow, which has a retrograde direction. That means that will move, in fact, against the movement of the tissue, and this will help, in fact, to produce this movement. So he will use laser dissection really as a complement to the model to see if the ring contribution is the only one, or if other contribution could be possible. But now I would like to be more critical about this laser dissection. It does a line. It's a line dissection. But use micro mirrors. They can be displaced, and it will just draw a line, probably with a software. I think it's own maybe commercial software, and it will just scan very, very rapidly on the line. So, but now I would like to look better at this movie, and I want you to be more critical because, okay, we see the relaxation, but it's very interesting that if we continue to look, we see also there is a recovery of the meshwork. And what is interesting to see that the recovery on your right seems to be much faster than on the left. So if one wants to be more critical, one would say that probably the meshwork here is not exactly the same as here. Like if the two experiments were not done exactly at the same stage, even though in the paper say they're done at the same stage. Okay? So what is going on? It seems that the turnover probably is much higher here. So this I just happened to be because I made up this movie, and by putting the two things together I could see this, because otherwise it's not trivial to see directly. So I was starting to think about this, and what I thought is that probably what is happening is that since you have much more relaxation here, probably you have much more depolymerization also of the actin, though probably you have a much higher local pool forming, and this will in fact allow the meshwork to form again, back again. Because normally these two are exactly at the same stage. So this could be one possible solution. If we think of, for example, Manuel Thierry experiments that he does, in fact he creates meshwork of actin on a petri dish, and then he puts myosin. This myosin contracts the actin, and this will create also a pool of actin, which create other fibers, because the actin in fact is getting depolymerized. So this is also something that could happen here locally. And that's why you have a recovery which is much faster here compared to here. The other possibility will be that this is also dependent on the way you cut, but then I will not know how to interpret this. I don't know how, maybe because the meshwork probably has some polarity also within, even though in this case it seems really quite random meshwork. Okay, then these are other experiments that I will just show. I will not comment too much because I will then present on the 21st, which is my new project. So I'm interested in a process called gastrulation. So in fact, it's the invagination of a tissue at the early stage of Drosophila development. And this is the ventral side of the Drosophila. This is what you're seeing here. What is marked is myosin 2. So what you can see is you have, in fact, a meshwork at the tissue level, at the apical region, which is formed. This meshwork has been shown already to play an important role for this tissue invagination. So what I do, again, with an infrared laser, femtosecond laser, what I try to do, in fact, is to dissect this meshwork to see how important is this meshwork to this invagination process. So as you can see, I can cut the meshwork. The cell, they do not explode. What they do, in fact, they start to enlarge. And normally these cells are apically constrict and start to go down. But what they do, in fact, they start to pop up. So they're no more constricted apically, but they enlarge. So what you can see with this experiment is that, in fact, this process is quite robust. Because even despite the great cuts, I cannot cut all the meshwork. I can cut a great part of it, but still the process is still going on. So this, I think, it will be the last example I will give you. It's something that I've been doing recently. It's a new way, in fact, of using laser to do other type of ablation. So this is called carbonization, and as you learned before, this is a thermal effect, laser tissue interaction. So what I do, in fact, I lower much the power compared to what I do when I do plasma ablation. And this, in fact, will introduce much more thermal effects. These thermal effects are very local. So what I managed to do is, in fact, is to carbonize lines of cells locally. So this carbonization will stiffen locally the tissue, but most of all, what it will do here in this specific case, in fact, it will attach the tissue locally to the vitelline membrane. So this egg, in fact, is within a container, which is a membrane, which is a vitelline membrane. And this vitelline membrane, during all the process of this tissue elongation and invagination, does not move. Okay? And the tissue will move within this shell. So what I'm doing is that by doing it superficially or at the apical site of these cells, what I managed to do is to stiffen these cells, but also to attach them to the vitelline membrane, which is a fixed object. In this way, in fact, what I can do, I can isolate mechanically cells within the embryo, or create walls. For example, if this tissue is flowing downwards, I can create a stiff wall and see how the flow of cells changes. So the experiments that I'm doing right now is the same, but I'm doing it not at the tissue level, but at the embryo level. So this is, okay, this is the thermal effects. And this is the example I'm giving you. You will see that the cells are blocked on one side, and on the other, they kind of flow around. And these cells seems to be stretched, like if it was like a really fixed anchorage point. So, as I was saying, I'm doing this kind of a study at the embryo level. And this is the kind of imaging that I'm doing right now, is by using a spin microscope, which is a light sheet microscope that allows, in fact, to visualize the full embryo in 7 seconds. And after that, you can do a time lapse. Okay? This way you have a visualization of the whole embryo. So what is interesting about these images, I'm going in Z, and you will see the good qualities on one side, but it's also on the other side. While normally when you have a confocal, you will see the image normally is degraded while you go in.